General properties of viruses, part 3. Cultivation of viruses. Viruses cannot be grown on artificial culture media like bacteria. For growing viruses, we require some live cells. And for this purpose, there are 3 different methods which have been used for growing viruses: animal inoculation, embryonated eggs, and cell culture. In animal inoculation, an experimental animal like mice, rat, monkey, rabbit, guinea pig, etc. are used for growing viruses. In embryonated egg has different portions. 4 parts of the embryonated egg are used for growing viruses: the chorioallantoic membrane, the allantoic cavity, the amniotic sac, and the yolk sac. The chorioallantoic membrane is used for growing herpes viruses, respiratory syncytial virus, variola virus, and vaccinia virus. And the allantoic cavity is used for growing the influenza virus, yellow fever virus, and the rabies virus. The amniotic sac is used for growing the influenza virus, and the yolk sac is used for growing viruses as well as chlamydia and rickettsia. Have a look at this figure. The embryonated egg, it has 4 portions which are used for cultivating viruses: the chorioallantoic membrane, the amniotic cavity, the allantoic cavity, and the yolk sac. Tissue culture. Animal inoculation, embryonated egg, and tissue culture are 3 methods for growing viruses. In tissue culture, again, there are 3 different types of tissue cultures: organ culture, explant culture, and cell culture. In organ culture, entire organ is taken for growing a virus. Example, for a respiratory virus like coronavirus, tracheal ring organ culture was used for coronavirus. Explant culture, fragments of minced tissue are used for growing a virus. Example, adenoid tissue explant culture used for adenoviruses. Then comes cell culture. In cell culture, cell monolayers are used for growing viruses. They are grown in cell culture tubes. Cell culture again is of 3 types: primary cell culture, diploid cell strains, and continuous cell lines. In primary cell culture, normal cells are used. They have a capacity for limited growth. Example, human embryonic kidney or monkey kidney. Then diploid cell strains, which are diploid cells, they can be subcultivated, serial passage for up to 50 times. Example, from human fibroblast. Then comes continuous cell lines. Example, cancer cells. Cancer cells, they can multiply indefinitely, so serial cultivation can be done on and on and on. Example, HeLa cells, Hep2 cells, and KB cell lines. So 3 methods for cultivation of viruses: animal inoculation, embryonated egg, and tissue culture. In tissue culture, again 3 types: organ culture, explant culture, and cell culture. Then in cell culture, again 3 types: primary cell line, diploid cell lines, and continuous cell lines. Now, when viruses are grown in cell culture, how do we detect that the virus has grown? What are the properties through which we can come to know that the virus has grown in the cell culture? Then there are different effects it can produce in the cell lines, like cytopathic effects, metabolic inhibition, heme adsorption, interference, transformation, immunofluorescence, nucleic acid detection, enzyme detection, and electron microscopy are various methods useful for detection of viral growth in the cell culture. In cytopathic effect, certain effect will be produced on the cell lines by the virus, and this effect on the cells can be seen in the microscope, and it is known as cytopathic effect. Then virus can lead to metabolic inhibition of the host cells, and this can also be observed by some processes. Virus can lead to adsorption of RBCs, which is known as heme adsorption. Viruses can lead to interference of other viruses in growth. So a known virus and an unknown virus, when taken together, the interference can be used for identification of a virus. Viruses can lead to transformation of the host cell genome, leading to cancerous change. Example, oncogenic viruses. Viruses can be seen in immunofluorescence in the infected cells. the effects can be seen. Then nucleic acid detection can be done for diagnosis of viral infections. Viral DNA or RNA can be detected by nucleic acid detection methods. Then enzyme detection can be done for diagnosis of virus. And electron microscopy can be used for observing viruses in the microscope. Cytopathic effect में here syncytium formation is shown. Syncytium formation in cell culture caused by respiratory syncytial virus on the left side and measles virus on the right side. Syncytium means what? Syncytium means cells adhere to each other, and this collection of cells adherent to each other, it is known as a syncytium. You can see in both these figures the cells have got attached to each other, and this kind of attachment is known as syncytium formation, which is one kind of a cytopathic effect observed in cell cultures infected by viruses. Here one more figure is shown in which a normal cell is shown in the microscope, and the normal cell line, once infected by the viruses, it will lead to certain changes in the cells, which is shown as CPE in the second figure, cytopathic effect. Heme adsorption is adsorption of RBCs. RBCs will get attached on the surface of the cells. Here in this figure you can see plenty of RBCs have collected at the site and they get adsorbed on the surface of the host cells. This is called heme adsorption, and it has been seen in influenza and parainfluenza viruses. Then inclusion bodies can be formed in various cells infected by viruses. Example, Negri bodies are found in nervous system cells infected by the rabies virus. Viral assays can be done for detection and quantification of viruses. Total viral particle count can be done by electron microscopy or heme agglutination. Infectious virion assay can be done by quantal assay or quantitative assay. The quantitative assay is again of two types, plague assay and pock assay. Plague and pock are lesions produced by the viruses, and one virus produces one type of lesion. So from the number of lesions we can determine the number of viruses present in the solution. Viral genetics includes mutation. In mutation, various kinds of natural mutations will also take place in viruses, and certain types of mutations like conditional lethal mutants and temperature sensitive mutants have also been observed. In conditional lethal mutants, these mutants they can survive in certain conditions, but in certain other conditions they get destroyed. So this is called conditional lethal mutants. Certain mutants are such that they can survive at about 20 to 22 degrees Celsius, while they get destroyed at about 37 degrees Celsius, and such mutants are known as temperature sensitive mutants. Then recombination of viral genetics can take place between two active viruses, between one active and one inactive virus, and between two inactive viruses. The gene of two viruses can get combined, and from this combination a newer setup of gene can develop which has features of both the parent viruses. Various non-genetic interactions have been observed in viruses. Phenotypic mixing, genotypic mixing, complementation, interference and enhancement have been observed in viruses. Now viruses can be classified in various ways. One taxonomical classification is dividing them into two categories, DNA virus and RNA virus. The DNA viruses are 6 families: herpes, adeno, hepadna, and Papua, pox and parvo. Herpes, adeno and hepadna, and Papua, pox and parvo. These are 6 kinds of DNA viruses. RNA viruses are about 13 in number: orthomyxoviruses, paramyxoviruses, Toga virus, Bunia virus, Flavi, Calici and Philo, Rhabdo, Rio and Retro, Erina, Corona and Picorna. So 13 RNA viruses: orthomyxo, paramyxo, Toga, Bunia, Flavi, Calici, Philo, Rhabdo, Rio, Retro, and Erina, Corona and Picorna. So 6 DNA viruses and 13 RNA viruses have been known to us. You can divide the DNA viruses and RNA viruses into different categories based on different properties, based on the type of DNA, double stranded or single stranded, and even in double and single stranded, enveloped or non-enveloped. Then examples of different categories of viruses include, like double stranded DNA viruses include herpes viridae, Papua viridae, pox viridae, and adenoviridae. And each of these categories has some examples given inside, like Papua viridae have polyoma and papilloma viruses. Pox viridae have orthopox virus, parapox virus, avipox virus, and molluscopox virus. Adenovirus has mustardinovirus and aviadenovirus. Herpes virus has varicella virus, cytomegalovirus, etc. Then RNA viruses can also be divided into single stranded or double stranded viruses, and again they can be further divided into enveloped and non-enveloped viruses. Enveloped single stranded RNA viruses include orthomyxoviridae, paramyxoviridae, rhabdoviridae, bunyaviridae, erinaviridae, and filoviridae. Orthomyxo, paramyxo, rhabdo, bunya, erina, and filo are single stranded enveloped RNA viruses. Double stranded non-enveloped RNA virus is reovirus. Single stranded enveloped RNA virus toga, corona, and retro. Single stranded non-enveloped RNA virus picorna and calici. Positive sense single stranded RNA viruses include picorna viridae, calici viridae, astra viridae, toga viridae, etc. And double stranded RNA virus is reovirus. Negative sense single stranded RNA virus include paramyxoviridae, rhabdoviridae, filoviridae, etc. Now, what are prions? Prions are proteinaceous infectious particles. They are further smaller than even viruses. मतलब वायरस स्मॉलेस्ट नोन है पर उससे भी छोटा है प्रायॉन्स, लेकिन प्रायॉन में कोई जीन नहीं होता, neither DNA nor RNA, and they are composed of solely of proteins with no detectable nucleic acid. They cause diseases known as prion diseases. Examples: scrapie in sheep, mad cow disease in cattle, Kuru and Creutzfeldt-Jakob disease in humans. We will study prions later on as a separate chapter. With this, we finish General Properties of Viruses Part 3. Thank you.